Why do we use MALDI? Because it's so fast. What advantages does it give us? The most important advantage it gives us that is increasingly recognized as highly significant is that proteins are not all the same. There are multiple variants of every single protein. In fact, if you look at the data, it's clear. The human genome is comprised of about 20,000 genes. The human proteome is comprised of about 1 million proteins. In other words, from every gene, there is a very large number of proteins that are generated from that. Those variants are detectable, quantifiable, and visible at the level of MALDI mass spectrometry. They are not identifiable by techniques such as ELISA. ELISA looks at the gross properties of a molecule, in essence, its shape.
So MALDI is actually really interesting because I think it gives us one of the most direct representations of the proteins circulating in your blood. A lot of times when people think of a protein, they think of a single thing. But what the information, because we don't do any chemical treatment of the sample before we analyze it, we really get a lot more detailed information. So each individual protein will show up in multiple different forms in the MALDI-TOF spectra. We see post-translational modification or different protease cleavages that give us a much greater diversity of the proteins that we're measuring. And so we're able to capture all that information and build it into the assays to provide relevant clinical information.
Where do you best fit? Do you fit best in the universe of control patients, or do you fit best into the universe of responders? And that's the strategy that we use. We're mapping a patient sample against two possible universes, the universe of disease or non-disease, or the universe of responders or non-responders. That doesn't require identification of those peaks. It says into which universe does the sample from this patient best fit.
In the broader community, typically people are trying to enhance the specificity of the mass spec method, and so they use very complicated techniques to get high resolution information on their samples. What is not commonly understood is that MALDI-TOF serum profiling, which is what we do, still gives us access to a number of clinically meaningful, especially in oncology, clinically meaningful biomarkers that we can measure directly from blood and get data on those biomarkers and then provide clinically meaningful information in a very simplified and compact workflow that eliminates the need for very time-consuming and complicated methods.
One thing that may not be so obvious is that by looking at a mass spectrum, you see contaminations, you see all there is there. So if you compare this to other protein measurements, you look for one molecule, you don't know whether your sample's been bad, you don't know whether it's been contaminated, or whether it's a donkey sample or a human sample or a plasma or serum sample. You have to deal with it. And so MALDI directly, just by the spectrum, you know where it's from. You get your quality control with the measurement automatically.
In recent years, the FDA has cleared two MALDI-TOF platforms for applications in microbiology. This, for the first time, opens up a hardware platform that allows patients to benefit from state-of-the-art technologies and help deliver new tests that help guide patient therapy.
What we do is interrogate the molecule at the level of its molecular weight, its composition. And so there's a dimension to the data that we get that's absolutely invisible, not apparent, not accessible by other techniques. The other thing that's important about MALDI mass spectrometry is that we interrogate all these proteins in one run. So if you wanted to develop an ELISA for one assay, that would mean one antibody, one test, one analysis, and then another antibody for the next protein. We measure hundreds of proteins simultaneously in one run by mass spectrometry. We measure all of the variants, well not all, but many of the variants of those proteins in that same run, and that gives us almost infinite complexity to the spectrum compared to those targeted assays that are based on ELISAs.